Welcome, class, to the second lecture on the immune system. I'm actually recording it before I give the first lecture to you guys, but I can keep that in my head. So we're going to continue on and get to the cool adaptive immunity. In the first lecture that you'll hear in person, I'll talk about the lymphatic system and then look at your innate immunity you're born with. And this is the cool adaptive, and it's a very complex subject, really. So, but I'll try to just break it down to what you need to know. And so, general terms, let's get right at it. Here we go. So, first of all, what are you afraid of? What your body is afraid of, really, is looking at pathogens. So pathogens are going to be things that cause disease. And I'm teaching this summer pathophysiology. So a requirement is A&P1 and 2. And where we go through specifically just all different diseases, everything. It's pretty cool. But let's take a look. What are you really worried about invading your body? What's out there, that's going to take hold in you and live in you and kill you? If it's a parasite, then it lives on you and doesn't kill you necessarily. It just kind of saps your resources. And in the world of diseases, really, the way to think about them is that they're organisms out there just trying to live and evolve and reproduce, just like you are, like squirrels and deer and everything else. So they want to live, and diseases want to spread. They have to spread, and they don't want to kill their host outright, because then they die with the host, right? And so they keep you sick, they keep you spreading it, and there's very few infectious diseases that kill you rapidly. They want you sick and spreading, coughing, and having diarrhea, and just, trying to find new hosts. And we'll see, once a critical mass of people can't get sick, get immunized, for instance, then it can't spread anymore, and thus, the disease isn't there. Otherwise, diseases just stay in the population, and they keep finding new hosts to infect. And on their end, they got nothing against you, really. It's just they're living their own good life, and they need you to reproduce. Anyway. So, of course, the viruses. We're here in COVID-19 as a type of coronavirus, and yeah, you can recognize there are other ones like HIV, one of the scarier ones. Herpes and measles and tetanus and Ebola. So lots of viruses out there. They're not technically life viruses. They can't reproduce on their own. They need a host cell that they invade, and they hijack that host to make more copies of themselves. Yeah, pretty tricky. And then bacterial diseases, all kinds of things you can think about. Strep throat and Salmonella, E. coli, tuberculosis, Lyme disease. A lot of bacteria that can live among us and cause harm. Of course, we have symbiotic bacteria that live in our guts, that help us out, live on our skin and other places in our body. Fungal diseases are pretty rare, called mycosis, it's a fungal disease, but yeah, yeast infections you can have, and ringworm, right, Canidia or thrush. And so there's a few things. Fungal diseases, but not many. Protozoans or eukaryotes, they're bigger than bacteria, right? They have a nucleus and all. And famously, we've looked at malaria quite a bit. Malaria is a protozoan spread by mosquitoes. Here in Maine, if you go camping, you don't filter your water, you can get Giardia, amoebic dysentery. You can get a protozoan that lives among you. And then bigger parasites, of course, bigger worms, tapeworm, roundworms, all kinds of worms you can get. And then strangely, there are proteins that cause disease. And these are the prion diseases, scary, but very rare. You get them, some from eating monkey brains, don't do that. But famously, mad cow disease And there's a chronic wasting disease. They worry a little bit. It's found in deer, and they worry in Maine. They has not been found in Maine, but they worry, you know, that it spreads, that it can spread to humans. But yeah, it's very little chance of that. Don't worry. But yeah, protein diseases are not even it's a protein that spreads in your body, a prion, and it changes the other proteins into this abnormal type, and can be some deadly diseases, but incredibly rare. All right, so pathogens out there, I mean, virus and bacteria are your main issues to worry about, causing a lot of the diseases that we think about. And we developed antibiotics against bacteria. Antiviral drugs are much fewer. Obviously with COVID, you know, common cold, things like that. Pretty much you just, you know, ride it out. You know, it's more difficult to combat this with a lot of drugs that we have. All right. So pathogens are disease-causing agents. I would say organisms. Remember, we talked about some are just proteins or viruses. And again, we have defenses to keep them, because they're all over our environment, to keep them from causing disease. And some things, as you know, you carry strep, you know, bacteria in your throat all the time. But sometimes when your defenses are down, when you're working too hard, you're stressed, they can come up again. So in many cases, we have disease-causing organisms that can only really take hold when your immune system is lowered for some reason. They can take hold. And others, like, you know, COVID-19, you need to be exposed to it, you know, and it can, depending on how well it travels, is how easily it's spread, of course. So in our defense against all these things in the world that would love to live inside us and make us their host, we have barriers such as our skin and the acidity of our stomach acid, and mucous membranes and the cilia in our throats that keep crap from, you know, accumulating in our lungs. So we have all these things that are there, and other innate defenses it means that you're born with them, innate. You're born, these are not specialized, but really rapid at killing anything that's foreign that's coming in and trying to prevent them. So in many cases, bacteria are stopped by our skin, right? But if they do get through there, then we have additional defenses where we have neutrophils and macrophages that could possibly eat them and take care of it, right? But let's say we have a major cut and there's a big invasion, and then we may have to step it up. And the adaptive defenses are your cool immune system. And these are ones came about later in evolution. Innate defenses, you'll find in fish and insects, stuff like that. Adaptive defenses is adaptive immunity that we're going to talk about. These memory cells and stuff is I believe it's just a mammalian thing. It came about later, and it's very specific and very cool. It takes a little bit of time to get going, you know, but once you're immune, like once you have a vaccine or something like that, you can be exposed to that pathogen, and it's just, your body quickly ramps up and destroys it. Yeah. It's that first exposure that takes time, you know, that you get really sick. But after that, it has that cool effect of making you immune to that pathogen in the future. All right, so when you hear innate, you think you're born with it. These are not specific against a particular invader, but any invader that is not self is attacked. And you take a look at this list. And I'll go through these, but just real quickly, species resistance means that, you know, you don't get kennel cough, right? You don't have to worry about, I'm going to give my cat my cold, you know. So the organisms are specific to certain species, and if you're not that species, that virus doesn't have, the receptor doesn't fit. Mechanical barriers, especially skin. I mean, your unbroken skin is a great barrier to things getting into you. Chemical barriers, talk about the saltiness of your skin, the high acidity of your stomach. And then you have a whole bunch of chemicals that we'll talk about briefly. We don't have time to get into detail in this class, but you have things like complements and defenses and interferon, things you probably heard about, that are chemicals that just are good against the bad guys, but they're not specific to any particular bad guy. We'll see we got a certain kind of lymphocyte natural killer cells that's really good at, beating up virus-infected cells. And inflammation, we'll talk about that. I've already talked about it. So when you get a cut or an infection, it gets red and swollen and painful. Of course, phagocytosis means eating, cells that eat, engulf the invaders. And then fever. Fever happens, your body temperature rises, and that's going to help against infections. But it's not specific. It happens. It could be any kind of infection. If you have a fever, it doesn't tell you, what kind of infection you have at all. All right, let's get to it. So first, species resistance. So indeed, that certain diseases are human diseases or dog diseases, right? Or cat diseases. And some things like, there's like some diseases will cross, like feline leukemia, or even some monkeys getting HIV. There's some that are more general diseases, but they're rare, usually diseases specific to a certain species. And it's because one can be the environment, so, if we have a disease like measles, it doesn't affect frogs or fish because their body temperature is usually so much lower. So it has to be a suitable environment for that pathogen. And then usually it has to do with specificity of receptors, a virus has to land on a cell. It's got to recognize the cell surface, some receptors, and then bind to it and inject its genetic material into that cell's machinery. And if you're a different species, that virus just simply goes over and doesn't stop. Indeed, yes. Oh, armadillos are interesting. They also get leprosy. Yeah, humans and armadillos. The hell does that mean? I don't know. Armadillos are weird creatures anyway. And you guys know they only have 4 identical twins. That's how they reproduce. Yeah. And when a car comes, instead of crouching down, they jump straight up. So down in Missouri, where I came from, Maine, they are the porcupines, of Maine, or armadillos are on the side of the highway everywhere because they get hit all the time. But we can study armadillos to look at leprosy as a model because they also, leprosy is in humans. And you can get leprosy from armadillos, but it doesn't happen much. And then I show this kid, I'm looking at the bird and the pig, and I just talking about disease, like in virus diseases and emergent diseases that are kind of scary, like COVID and others. They come from, the virus was in a different species. Like surely COVID was in a bat. I mean, bats and birds have a lot of diseases, a lot of viruses, and usually they don't affect humans. But you'll get a situation where there's contact between these. And sometimes a pig too is this intermediate kind of mixing vessel between bats and humans and other things. And if the virus mutates enough, so all of a sudden it doesn't just recognize bats, but now it recognizes human cells. If there's that change, then for that virus, it opens up this new host and it can spread. Pangolins. This was a earlier slide, and I think they've been kind of crossed off the list. We don't know. Looking at the host just last week, I mean, there's news about going to Wuhan, the market, to see what kind of animal COVID spread. It didn't spread from a lab. It's like the evidence says no. It didn't escape from a lab. It came from a natural host out there, but they don't know. It says badgers and ferrets and this pangolin are the suspects. Looking at where was this jump between the animal and human. That's the big danger. And COVID is the one that we're talking about now, but realize this danger has been talked about for years and years. It's just a danger is that in a pandemic, I mean, you see what COVID did. And then if if HIVs mutated so it was easily transmitted, right now it's kind of hard to transmit, you need fluid. I mean, it would be incredibly dangerous. And so as humans go into rainforests and destroy ecosystems, we have more contact with organisms, other mammals that can mutate. So anyway, this is COVID-19 shows what can happen, and it's been a year now when the virus switches from other animals to humans, and it's going to happen again and again. So we just need to get better at this and make sure we're ready. All right, so barrier, so species barrier, that keeps a pathogen from just quickly being able to go to humans or other animals. Now, mechanical barriers are just great. Like your skin is a great defense. I mean, for one thing, you remember how skin grows from the inside out. So even if bacteria try to take hold on your skin is sloughing off, right? And so it's moving out. Anything's got to like go against the current. It's going to try to get in through your skin. Now, if you have cuts in your skin, cracked skin, things like that, then it gives a better entrance to pathogens. But unbroken skin is a beautiful barrier. But where there's some breaks, your mouth, your nose, your eyes, your genital tract, your inside your trachea as you breathe in air. So there are, of course, there are dangerous areas, and that's why we worry about COVID spreading if you touch your T-zone here anywhere. And here you could take that virus that can't get through your fingers, but then you put it in your mucous membranes and it can gain entrance to you. So just a good example of how skin is a good barrier. Yeah, and your salt itself, as you sweat, it gets, did I say salt? Your skin itself, as you sweat, it gets salty. And of course, salt doesn't allow bacteria to grow. Before refrigeration, we salted things or dried them out, beef jerky, or we salted fish and things like that. Keeps it from going bad. And a whole bunch of chemicals that this is non-specific that can come out. In your tears, there's lysozyme. That's an enzyme that breaks down bacteria, and your constant blinking in tears keeps things from growing on your conjunctiva of your eye, right? Interferons, there's many different kinds, but you'll hear this too in medical shows, things like that. So interferons are given, and they're kind of non-specifically keep the cell machinery from making it easy for viruses to reproduce. So if a cell is infected by a virus, it will give off interferons, and the neighboring cells will notice that, and then they will take steps to make it harder for the virus to reproduce inside them. And so, so interferons, defensins are given off. I'm just going to name a couple of these because they're in your book, and they'll be given off, and they help destroy microbes. And then complement. If you get into immune, you take class in immunology. Complement is a whole bunch of proteins, and there's a complicated cascade of reactions that takes place in complement. But it's these little group of proteins that's important in helping fight infection, and they will call the cavalry in, and they will make inflammation happen. And so, a whole bunch of chemicals that are going to be involved in non-specifically just helping start that fight. All right, natural killer cells, NK cells. Now we're talking about lymphocytes in a moment. It's going to be the B cells and T cells. And those things are specific, smart cells. Now, there's a group of lymphocytes called natural killer, and they are not specific, but they look for cells that are different, that need to be destroyed. And if a cell has cancer, that means it's a weird cell, and it's probably displaying things that are on its surface that are not normal. And same thing if a cell is virally infected. Well, that can be a problem for your immune system, because if it's something inside your cell, if the outside of the cell appears normal, you're not going to be able to attack it from the outside, right? But virally infected cells are going to have unusual stuff inside of it, proteins and RNA and stuff. And so they're going to look a little bit different. And these natural killer cells, they look for cells that don't belong. Something is wrong with them, and they'll come up to them and they'll blast holes in them chemically. They'll shoot holes in them and or give them a signal to tell them to kill themselves. So natural killer cells, non-specific type of lymphocyte. So inflammation. And I've been talking about that in this class earlier, but you know what inflammation is, right? You've all had cuts, scrapes, splinters, where it turns red, painful, swollen, warm, right? This is all sounding familiar. Yeah, it could be a scrape and it turns red, or it could be some effect. Here's my thumb. It's infected, antibiotics. You need antibiotics if it gets that serious, or sunburn, something like that. And what's happening is that in localized area, you're having histamine and heparin coming out. Blood is being drawn to the area, carrying with it the cavalry, the neutrophils, and eventually even clotting factors. You kind of make a fibrous kind of shell around the infection to keep it from spreading. And then over time, you break that down when you're healing. But if you've got a cut where you've let in some bacteria, they start dividing inside of you and eating you and using your energy. You've got to beat that, because if you don't, like in this case, without antibiotics, that infection will keep spreading and it can be life-threatening. So normally your body can take care of it, but we got some antibiotics that work against bacteria too. So inflammation is a process, non-specific. If you have an infection, you have chemicals that cause this increased blood flow and possibly a fever if the infection is big enough. And it's the way your body reacts to any kind of invader. So that's why it's innate. Phagocytosis is cell eating. And the main ones are, of course, neutrophils, which make up like 60 or 70% of your white blood cells. Neutrophils are the cavalry. Again, they're dumb. They come, they see something unusual. They can eat 10 or 12. They blow up and turn to pus. But yeah, we need them. Neutrophils are fast, and they're going to eat the bad guys. And then macrophages. And in our blood smear, they were monocytes. But when they're going to turn into macrophages, and as they get into the tissues, you got macrophages everywhere. And they're bigger. They can eat 100 bacteria, and they can eat bigger bacteria, and they can do some other cool things. And so you'll see those more kind of in chronic long-term disease, as opposed to neutrophils, are fast and they get there. Yeah. But both these guys, the neutrophils and the macrophages, are the big phagocytes. And some others can do it too, though. And this fever, the fever used to be thought, oh, Jimmy's got a fever, we got to bring it down immediately. And now we know fever is a response. It's not a symptom to be combated. If the higher body temperature is not so great for microbes, they prefer more body temperature. So you're doing that. And also we know that with a fever, you sequester iron out of your blood, and you keep it in your liver and spleen. And bacteria, a lot of them need iron to reproduce. And so with higher temperatures, it's worse for the bacteria. Now they don't have as much iron to eat. And then it kind of ramps up your immune system. So a fever is something that's part of this innate immune system, and it's a natural thing. Obviously, too high a fever can be dangerous. You have 104, 105, you can have permanent damage, right? But a normal fever, you'll just see it. It's part of the innate immune system. It helps. All right, now let's get serious. So all these things, fever, inflammation, being a certain species, all these things will help against attackers. But now we're going to talk about specific immunity, adaptive. So you're not born to this. This comes about from living a life in this world filled with all these organisms. All right, what's critical is that you distinguish between self and not self invaders. And we'll see when this breaks down, when your immune system starts recognizing your own cells, that's an autoimmune disease. And then you've got MS or rheumatoid arthritis or lupus. So autoimmune diseases are bad when you recognize yourself. But immunity means you recognize foreign invaders. And it can be bacteria, viruses. It can be toxins as well. We'll see organ transplants too are foreign, and so sometimes you'll fight against those. We have to battle that one. But again, what's most important is that your immune system does not recognize any of the molecules that are normally in your body. So I have that picture there, woman looking in a mirror. So early on, your immune system is trained to not fight Jeff's cells, recognize Jeff's normal cells. Do not attack them, because if they start mounting an attack against my spleen cells or my myelin sheaths and my nerves, then it's not good at all. All right. So early on, you're before birth, you take inventory of every single molecule, not molecule, every single surface molecule, I guess, on all of my cells. And any of my immune cells that want to attack myself, they're done. We just keep the ones that don't recognize self. Okay. And this just to remind me that sperm and eggs could cause a problem, because sperm and eggs are they're haploid, right? And they're different than us, right? They're made through meiosis, and so they have a unique combination. But sperm and eggs are protected. Your eggs will be in this follicle, they're protected, and sperm in the testes, there's this blood testes barrier, so we don't attack that. And then the fetus of a pregnant woman too is different genetically too, but that's protected by the placenta, so there's not many antibodies are going to cross from the blood to the placenta. All right. So what are we attacking? Those are called antigens. They're any substance that causes an immune allergic response. All right, so something different. And it turns out the best ones, the ones that are most effectively an antigen are more complex. Simple, H2O or something is not going to be one, but any kind of complex molecule is going to cause, you're going to recognize that as different. Your haptens are small molecules that don't elicit a response, but you put a couple together, then they do. So a few cases of those, kind of cool. So antigens can be proteins, glycoproteins, sugars, any kind of different molecule. When I say the more unique, the more response it has. And we learn antigens because of the blood types, right? You had A or B antigens on the surface, or both or none. Right? So something surface molecule. A bacteria gets in you, it's going to have all kinds of surface chemicals, and then something that you'll recognize as not normal, not self. All right, so lymphocytes are white blood cells, and all blood cells are made in your bone marrow. So these are all coming from your bone marrow. And the deal is the T cells are going to leave your bone marrow and go in your bloodstream, and they'll make their way to your thymus gland, not thyroid, thymus gland. And the thymus gland is where they will mature into T cells. That's where they get their name, thymus-derived lymphocytes. And then the B cells, the other big lymphocyte, they just mature in your bone marrow. You can remember B is for bone marrow, but it wasn't named that way. But so B cells mature in your bone marrow, T cells mature in your thymus. They're both made in your bone marrow. That's where they mature. That's where they are tested to see if they recognize self. And if they recognize self, they're dead. All right. You only want ones that recognize non-self. And then once they mature, where are these lymphocytes found in your body? Well, we found them in your blood. Remember, took a prick of blood. Oh, look, lymphocyte, right? But they're not doing much in your blood. They're just going somewhere. They're going to travel in your blood. They're going to leave your bloodstream, and they're going to hang out in your lymph nodes, your tonsils, your spleen, your appendix, all over your body. They're going to hang out, these lymphocytes, ready to look for foreigners, invaders in your body. All right, well, the cool thing is, oh my God, wait till you hear this. Your B cells and T cells, these lymphocytes, you have millions of varieties, and each different one has a unique signature on it that's looking for a unique chemical that matches it. I know. So each one of these B cells or T cells have thousands of these receptors on its surface, all the same, but each one is unique. There are populations of the same ones, but there are millions of unique varieties of these lymphocytes. And you wonder, because normally when you divide cells, they're identical, but they do it through shuffling of the genes. And so early on, before you're born, you make millions and millions of varieties of lymphocytes. And each one is ready to recognize some different combination of chemicals in the world. And a whole bunch of them that would recognize yourself are destroyed. So hopefully all you have are these millions of varieties that are ready for any kind of weird chemical, anything that's foreign that's not yourself. Picture that right now in my lymph node. Feel my pulse, but my lymph node, I've got all these lymphocytes, B cells and T cells, and a foreign invader comes by it, and it just bumps into thousands and thousands of them, And that chemical on that invader is going to fit in one of these millions of varieties. And when it fits, that lymphocyte wakes up and it's time, it's go time, right? So that's the deal. You make all these varieties early on in life, and then they just wait. Most of them die with you and they've never been used. But the one for chickenpox, the one for COVID, anything, might come to be very, very important to save your life. All right. So now we're talking about this cool, as you can see, the lymphocytes are going to be your adaptive immunity acquired. So this is where you recognize something in foreign, and then your body adapts and gets ready to fight that specific invader. All right. So the antigen is going to be the foreign substance, probably a molecule on the surface of a bacteria or a virus. It has to be recognized by the particular lymphocyte that fits, that has that combination that fits the chemicals on the surface of that invader. And once it is the chosen one, once it fits, that cell gets excited, activated, and it will start, it'll make millions of clones of itself. It will divide like crazy. You're going to see it. It takes a couple of them to help each other. And but once that happens, once this new invader has reached your body and those lymphocytes that are in question are activated, then they go full on to fight that invader specifically. So not like a fever, not like a neutrophil, but this is antibodies and cell to cell that are going to attack a specific chemical signature of an invader. So the first step is recognizing. You got to make sure that that invader is made, the parts of it are made, presented to the lymphocytes so that they can recognize it. And it's super specific. It's got to be the chemical signature has to match the chemical signature on that lymphocyte that surrounds the lymphocyte. And then the cool thing is that the next time you're exposed to that, you don't get sick. Your body just ramps up that response within hours instead of weeks. And so you don't even know you're exposed to it probably. All right. So it turns out that that foreign invader with a particular chemistry, what it needs, at least for the T cells for sure, what it needs is the T cells need to be presented with it, like this waiter needs to present it. It can't just find it out there. It wants to be shown this little piece of the chemical. So we call these antigen-presenting cells. And these will be macrophages and some other cells that they will, let's say they're going to eat the bacteria. They destroy the bacteria and they take pieces of it and they put it on their surface. And then they hang out, or they'll move through a lymph node with this piece of the bacteria on their surface on a particular protein that says, dude, I'm showing you this little piece that I found in this invader. And then it's going to bounce past all the B cells, the T cells, until one of them says, it's going to stick to it. It's going to be like, oh my God, it's me, you know? And so antigen presenting cells, pretty cool. And so, of course, these are everywhere and they're destroying bad guys, but then they take the pieces of the bad guys and they present them on their surface. Now I'll introduce this to you, this major histocompatibility complexes. I don't know, you're probably not familiar with this, but there's one of them. The MHC2 is where a little protein that's going to display the antigen that it found. And MHC1 is found on all of your cells, except red blood cells, but all the cells that have a nucleus have MHC1 on it that's unique to me, Jeff. And they'll do research with meadow voles and mice, and they'll see the more closely related they are, the more closer their MHC1 is. And so they know to mate not with close relatives. Yeah, so if you get into this kind of thing. So MHC1 is found on all your cells. That it's going to just show that it's me. And if you're virally infected or you've got cancer or something like that, it'll screw up that MHC1. It won't look like you. and then you'll be noticed and killed. But here we're talking about MHC2 molecules on the surface of these antigen-presenting cells, and that's where these T cells know to look. If they see that MHC2, they know on the surface of it, it's displaying something that it found. That antigen-presenting cell found this bacteria, and there may be a whole bunch of chemicals on it, and it's going to present little pieces of it all over itself. Right. So this just showing, it looks a little complicated on the slide, but see all those purple? Those are all lymphocytes. Just millions of them, right? And the antigen-presenting cell with that antigen is going to just be in contact with all these. And maybe only this one is the one that's going to fit. And if it fits with some disease, you get chickenpox, something like that, it's going to make just millions of clones of itself, as it takes a while to ramp that up. But that's the deal. Hopefully I get that through, is that, believe it or not, we're born with, there's millions of varieties, and it's just every disease has a little different signature on it. It's going to wake up one of these, and I'll show you what happens. And one thing, of course, we think about here, as I just mentioned, is that on one particular bacteria, it can have a dozen different kind of surface molecules. So you hear about monoclonal antibodies or polyclonal. So you could have make antibodies for one particular, like the spike protein on COVID, make antibodies. That's cool. It's going to kill it. But if there were several different proteins on it, you could make several different kinds of antibodies even better. So when you hear that. And right now, monoclonal antibodies, at least in Portsmouth Hospital, is what they do with people that are on COVID now, that are high risk, older. They'll give them these antibodies, and it will hopefully bind to COVID that's floating around and take it out. All right, so I talked about cells kind of just being excited and activated when they have a particular antigen here. Now, T helper cells, it turns out, we talked about B cells and T cells, right? And I'll get to exactly more what they do. And then T helper cell helps both of those to really get excited. I mean, T cells and B cells can recognize a foreign antigen, and they can get excited, but they really need that T helper cell. It gives off chemicals, these cytokines or chemicals that will get them excited. And so it's like the middleman that is necessary to have a real reaction take place. So just trying to review, this antigen presenting cell is going to eat something and then display on the surface on the MHC2 is going to display a little piece of that invader. The T helper cell is going to notice it, and it's going to say, oh my God, it's me, I fit this. And so it will start cloning itself and dividing like crazy. And then it'll also give off chemicals as T helper cells that will activate B cells and T cells that are also a little excited, but they're really excited. The T helper cell gives off chemicals. And so like all three are in agreement. Yes. All right. We all agree this is a foreigner. We all agree we're the ones, and they're going to divide like crazy. And so you may say to yourself, why do you need T helper cells, or why do you need both B and T cells to be? Why can't just B cells be activated and make antibodies? We don't actually know the exact answer, but in my mind, I think it's kind of like a safety feature where like having a committee instead of just one person making decisions. And it's if they all agree that this is something foreign, then they go for it. But again, hypothesis. So I mentioned cytokines real quickly, but these are chemicals given off by these cells that have various functions. And one of them is to get T cells and B cells excited, right? But you can see interferons I talked about are types of cytokines. Yeah. And the deal is you've got millions of varieties of these B cells. And one of them, in this case, this one, is a match. And so that one is excited. And then if it has cytokines from helper T cells, it starts this clonal reproduction, divides in two, the two divide into four, and it just goes on and on and on. And so you've got this it was a selection of that one. It's going to make clones of itself. Just tons, like a huge army is going to build very, very rapidly. And the first time me telling you this is that B cells turn into plasma cells. Plasma cells are antibody machines. They split out, honestly, thousands of antibodies per second. And antibodies, these little violet-shaped things, are just proteins that will stick onto invaders. I'll get to it in a minute. And then also, you make some memory cells, some B memory cells, and they will reside in your body the rest of your life. Them or their progeny, but a group of them will always be, after every disease, will be there. How cool is that? That's the cool memory part of it, right? And so once you get this disease once, you had you got sick, you made the impression, you made the attack, and then you'll have memory cells so that next time they wake up quickly. There isn't that big delay. You don't have to go through all this business again. All right, so there's two kinds of attack really here. We're going to talk about cellular immunity. So it's cell-to-cell combat. The other one is using antibodies. So cellular immunity, is you get these T cells. You'll hear cytotoxic T cells, which means toxic to cells T cells. So they're going to destroy cells, kind of cell-to-cell combat. And so a cytotoxic T cell has to recognize that a cell is virally infected or cancerous. It will come up to it and it will send out chemicals that will destroy it. So that's why it's called cellular immunity, because the cells. Yeah, so it's really good, like I say, virus infected or cancerous cells that are, because it goes right to the cell, as opposed to just, a virus or bacteria floating in the blood. So here is a cell and it's punched this hole in it. Below is the cytotoxic T cell. It's killed that one. Here's another picture. That's a cancerous cell above. Like David and Goliath here. That little cytotoxic T cell has shot out these proteins, these perforins that perforate it, make holes in it, and it just, everything leaks out and it dies. Yeah. So that's cellular response. The T cells do that. The B cells, we call it a humoral response. And your humors are your body fluids. So what happens in the body fluids, and it's those B cells that turn into the plasma cells, which are type of B cells that make antibodies, antibody machines. And these antibodies are proteins made by these B cells that just go out into the blood. So the B cells don't fight other cells, cell-to-cell combat. They just sit back and they have been programmed to make a specific antibody that the chemical is going to fit on the invader. And so that's really good at destroying things that are kind of in the fluids, you know what I mean? Instead of like a virally infected cell, it's already in the cell. So that's going to take cell-to-cell combat. But something that's floating there, like a bacteria, a toxin, a virus, the antibodies float, they're going to stick to it. Yeah, so this kind of shows you above of what the antibodies can do. So imagine this virus. Imagine it's COVID and it has spike proteins on it. And the antibody fits the spike. And so here's the virus, with its spikes, having a great day. It's on its way somewhere in your body, right? And then here's some antibodies that find it, they're going to stick to it. Oh no, I got an antibody on me. Another one. And they're going to stick all around it. And if you have antibodies on the virus can no longer reach the host cell. It can't, and then it's useless. It'll just be destroyed. And other things that they can do is these antibodies can cause a whole bunch of viruses to stick together to this big clump, something called opsonization, where it makes the virus more visible so that macrophages can eat it. That's why I mentioned, like you tag these trees with a tape or a spray paint that tells the log cutter that these are the ones that are destined for destruction. Well, antibodies will make any kind of invader more obvious to be eaten by the macrophages too. Yeah, there's just some cool pictures. It's showing, here's a cytotoxic T cell, and then these red granules are these little chemicals that are released, and they're going to cause that cell on the right, which has a virus in it, to be destroyed. So this is cell response. The humoral response is just using antibodies. All right, so here's these B cells and type of lymphocytes that when a particular one is picked, it turns into plasma cells, which will make antibodies, and then memory cells too. And they just hang out for the next time. And then also lymphocytes, remember you have cytotoxic T cells. So these have been primed to recognize cells that are invaded or bad, and they will destroy them, cell-to-cell combat. And then helper T cells, remember we talked about that, they're kind of in the middle. They are activated by the antigen, and then they send out chemicals that just help, that they just excite the other two. Yeah. The natural killer cells are a little different. They're not part of the specific immune system. They look for anything that's bad and they kill it. All right. Oh, I love it. So this is very helpful. Take a look at this. You see humoral response with the B cells and cell mediated there with the T cells. All right, I hope you guys are hanging in there. I guess you can pause. I've been going a long time here, but I can do it. Yeah. So at the center, here's the antigen. And I hope you all know that's something that's foreign that's going to be recognized and cause this response. That's the bad guy. And it has to excite all three, right? B cells, T cells, and helper T cells. And then the helper T cells in the middle, what are they going to do? They're going to give off cytokines, these chemicals that are going to excite those two that have already been primed. And it kind of gives them permission to divide like crazy. And then you got huge armies that are forming from this original one that is the chosen one that recognizes the bad guy. And so the B cells make plasma cells that make antibodies that go out into the bloodstream, out into your fluids, and they find bad guys and stick to them. And memory cells. The T cells are going to make these cytotoxic T cells. They're going to go up to cells and attack and destroy them. And then there'll be memory T cells too. So once you have this response happen, you've got memory B and T cells that reside in your body, and they just, rest of your life, you got them ready to go. All right, we can talk antibodies. You can see they're not cells, they're just proteins that are made by these B cells. And they have, just anatomy-wise, they've got two big heavy chains and then two smaller ones. So they're put together like this. They're very small. They're cheap to make, but they're just going to be released and they float around. And the interesting part isn't this bottom. The interesting part is the top. That's going to be the variable region on top, and that's going to be specific. So if you got a COVID virus with a spike protein, antibody is going to have the opposite to that spike protein. It's going to stick to it. Very specific. It's not going to stick to anything else but that spike protein. Yeah. So you guys realize this, you can have your own immune response and make your own antibodies, but you do realize you could take somebody else's antibodies, right? And give them to yourself and they'll still work against the virus. That's true. We'll talk about that. But normally if you're infected, you're going to make your own with your B cells. And so what do? What do antibodies do? Now you know that, I mean, we didn't get to the chemistry of it, but you need to know they're Y-shaped, and this is the variable end on top that's specific to a specific antigen. Well, they're going to stick. They're going to stick to the surface of any kind of not only bacteria, virus, but a toxin, a venom, something like that, right? And so they're going to cause it to agglutinate. They're going to make them stick together. They're going to precipitate them out. Or imagine you got some kind of toxin, and then it's going to neutralize it. If it's the toxin does damage, now stick around a bunch of antibodies around it. It can't do its damage, right? Nice. Yeah, cool. Complement. Again, the word opsonization means it makes the antigen more obvious to the phagocytes. It kind of like marks it. Definitely. Yep, and antibodies also work in promoting inflammation, all these things, but pretty much, that's awesome. You have antibodies that are very specific, and you've got just, imagine one B cell puts out thousands per second. So imagine how many antibodies you have. And if you have exposure to some virus or bacteria, and you have thousands of these, millions of these antibodies, they will stick to that invader and how quickly they can neutralize it, right? All right, well, what happens when it goes wrong? What if all of a sudden that you recognize your own blood cells as being foreign and you start to attack them? Not good, right? So autoimmune diseases, and you're probably familiar, could go through a huge list of them. And it's your body immune system gone awry and attacking itself. Type 1 diabetes, you attack the islets in your pancreas, you don't make insulin, right? Well, for instance, multiple sclerosis, MS, it's really a mystery. It attacks your own myelin sheaths in your central nervous system. And so what happens is that you probably have a different gait, maybe your vision's affected, things with your central nervous system, and then it's kind of hard to diagnose, but they can look and they see your myelin starts degrading. It turns into scar tissue, and then you can't have signals, be traveled by these nerves. So very bad. And so what do you do basically with autoimmune disease, rheumatoid arthritis, MS, lupus? You can treat the symptoms or just try to lower your immune system so it doesn't attack itself. But any kind of like steroids or drugs that suppress your immune system makes you more susceptible to other diseases, right? So you've got to weigh, the benefits of keeping your body from attacking itself to allowing it to still attack, all the foreign invaders out there. Yeah. MS, I guess we talked too. Do we talk about it in nervous system? We might have, but it's a weird one. You look at most infectious diseases and look on the epidemiology of it. It's found in the tropics. A lot of infectious diseases because there's a lot of diversity of life there. But MS, there's a gradient. It's greatest in Canada and in Minnesota and northern United States, and less in the South. And you see that. Look at that in Northern Europe too. The cold induced? Could it be vitamin D? Yeah, that's actually thought it's possible, right? But maybe it's environmental. Maybe it's some infectious disease, some virus or something, probably not genetics because there's a lot of mixing. So there's something in environment. Maybe it's an infection that further turns into an autoimmune disease. And we think that's maybe how some autoimmune diseases happen, is that you're fighting off a particular virus or bacteria, and perhaps the molecule on the surface of this virus is similar to a molecule on our own myelin. And so you make antibodies against this virus and you get rid of it. All of a sudden, some of these antibodies are like, hey, this looks familiar, and it attacks your cell. So many autoimmune diseases we see possibly start with an infection of a different kind, and then your immune cells are tricked and they start fighting your cell. That's one direction. It's a really interesting subject. All right, this really shows you, I love this. So I'll tell you what it is first, I guess. The y-axis is the amount of antibodies that you have. So look at the first time you're exposed to a disease. Look at how much time it takes. Those are weeks. I got like 2.5, 3 weeks before you've got, that much antibody in your system. And then you can see, you fight off the infection, right? Or you die, Usually you don't. On the other hand, imagine you're hit with it again. You're like a year later. Oh my God, look at that. Within hours, within days. So we're talking about in 3 days, you've got more antibodies that it took you 3 weeks to make. And look at how many antibodies you make. So that's the key to getting vaccinated, to being immune after that first exposure to a disease, is that you are primed. You've got B cells and T cells that are already, they remember the last time this happened. And if they get excited, they just pump out the antibodies so quickly that you may be exposed to something. You may be infected actually, but you very quickly destroy it all and then you're not sick. That's the beauty to our adaptive immune response. I'm sorry, that's just so cool. And then B shows like a different antigen, different disease. And you can see same slow response. But once we've been exposed, either naturally or through a vaccine, our next exposure is so fast and so overwhelming that we don't even get sick. All right, so the primary immune response is our initial exposure, and it takes weeks till we build up these antibodies. So adaptive is slow. Your innate system is quick. You're ready to go with a fever and neutrophils. But to make this response, you can see it takes a while to build up. But you're ready the next time. That secondary response is much faster and within hours instead of days and weeks. Yep, because you have those memory cells that sit with you. And we do talk about increasing allergies and asthma among children today. And some of maybe that were not exposed as children. It's called the hygiene hypothesis. Exposed as children to a lot of dirt and bacteria. We used to live on the farm and dig in the dirt. Now we have antibacterial soap, antibacterial toys. We're not exposed to as many animals and things in dirt because parents are too clean. And it's been found that kids in households without pets or that are homeschooled and not exposed to a lot of people are more likely to have these autoimmune issues, these allergies and such. So again, getting dirty, exposing yourself is gonna prepare you for all these different invaders later in life. All right, this is just a slide that is just a review again, and you can see all the variety of B cells and T cells you have. But when an antigen comes in, an invader, only one of them, could be this one, could be Charlie, could be Fred, could be Sue, but it's this one. That one gets excited, and that is going to make clones and clones of itself that will make these B cells that will make all these antibodies that can kill the invader. All right, we're getting near the end here. What about these diseases that we, oh my God, we used to suffer through so many childhood diseases. We're talking, it's not until like the 1960s that kids got measles and whooping cough, and before that smallpox, and all these mumps and all of rubella, all these diseases would really take a toll. And that's why actually life expectancy has gone up so much, not because we've prolonged life and that on average, we've stopped these childhood diseases. And yeah, I got chicken pox, me and my brother, when we were kids. Flying down to Brazil, we got it. I remember we were in a hotel room with chicken pox. It sucked, like for a week or so, but then we got over it. And you guys will never know that the horror of chicken pox and the scratching and everything, but because you have vaccines now. But I wouldn't get it again. I knew that. But then I could get shingles later in life. It's the smallpox virus just resides in your cells. It can come back in your old age. I'm too young for that, but I just had a vaccine, the Shingrix vaccine, so I shouldn't get shingles too. So anyway, so what's with these diseases, and why are some we can get a vaccine? Well, why can't we get a vaccine for the common cold or for HIV? Why is the flu vaccine? We need new varieties every year. This COVID vaccine, is there fear that new varieties are going to make our original vaccine useless? Well, it's really great stuff. And it has to do with things like mumps. They're a very stable disease. The DNA is very stable. Measles, a lot of, I can't really get into it too much, and so our vaccine can be very useful and stable. But the common cold and HIV, it keeps changing its face and evolves so rapidly that we need a new vaccine every year to kind of anticipate what flu strain is going to be here. And it has to do with really sloppiness of copying. Making mistakes is good for the disease because it can evade our modern day vaccines. Anyway, another thing I want to mention is that a lot of our really bad diseases come from animal vectors. And so chickenpox and smallpox, we think came from cowpox. And we can look back at a lot of our diseases now with people living in close proximity to our livestock. Look at a lot of Asian countries, there's chickens and ducks that live right under the house and in the house. And in the Middle Ages, we lived with our cows and pigs in the same room almost, I think. I don't know. So this close proximity allowed viruses to mutate. And a lot of our diseases jumped, like COVID, we believe, and HIV, jumped from animal to humans by mutating. All right. All right, so for instance, measles. When I was born, you can see there was, we don't know when I was born, but hundreds of thousands of cases every year of measles. Unfortunately now, it's going up because people have this fear of vaccines and really kind of this stupidity, if you excuse me, I'm not going to apologize, I'm going to tell you, of not wanting to get these vaccines. I'll talk a little more about that, but you can see what the measles vaccine did. We figured out a second dose is nice after that, but now it shouldn't be a problem in the US. And if you look at here's pre-vaccine amounts of people that had disease, and then since we had the vaccine, yeah, right? Smallpox has been eradicated from the world because of the vaccine. It exists in the CDC, and then in Russia, we think they have some. So right now, smallpox would be a huge bioweapon because you guys are all, we're all have not been exposed to it, unlike your grandparents and such. Yeah, and you look at polio. We had hoped to get maybe eradicated. COVID kind of slowed it down, but polio only exists in Pakistan and a couple other countries. That's it. But we have a vaccine that, again, both my mom and my stepdad had polio as kids. It was a huge fear and it killed so many people. But now we've almost eradicated it. And you can look at all these things. So vaccines have a lot to do with why we don't have childhood diseases. And I'm sure all of you have your tetanus shot, right? Vaccine. Well, I'll just mention briefly with this, talk about, and it's so important today in our news, the anti-vaccination people. And it all really began with this guy, Wakefield, who is a doctor who published a paper saying that the MMR, mumps, measles, rubella vaccine you get as kids could cause autism. And that's where it started. It turns out it does not. I'm telling you that, period. It does not. So many studies with hundreds of thousands of people, there's no link whatsoever. But turns out, we found out later that his study was falsified. He only had like a dozen people he cherry picked, and he was getting paid. He was getting paid to have an alternate to the vaccine. So he lost, the papers retracted, he lost his medical license in the UK, but still he still goes around as if he has any credibility on this. And of course he doesn't. This push with people that are fearful of vaccines, and like the COVID vaccine, we don't know what's in it. We know exactly what's in it. There's a list that comes that shows you exactly. I mean, because you don't understand what's in it doesn't mean we don't know what's in it. And I digress. But so what we have here is an outbreaks of measles and mumps and whooping cough all over the place that should never be a problem now if we're vaccinated. But we have people with this fear and this idea about freedom about this that is causing these huge problems among the anti-vaccination crowd, which some of you, well, you all know of people that are like this. Yeah, and this Reject Big Pharma was it last year, a year before? And it turns out it was nothing about Big Pharma. It just had to do with people not wanting vaccine, the vaccine deniers, because Big Pharma doesn't make big money on your measles vaccine. It was just people that were going under the guise of it's rejecting Big Pharma because that seems kind of popular, right? And Big Pharma I'm married to a pharmacist, but has big problems. Money making. We have a money making system for it, but vaccines are not part of that. All right, so immunizations. I mean, of course, cool story how they took the pus from cowpox and put it in a little wound, and the people wouldn't get smallpox. And so how we figured out we could do this, and what we simply do is just give a weakened version of it, or a part of it, or a dead pathogen with the antigen on it, and it will cause the immune reaction without you getting sick because it's weakened or dead, or it's just a part of it. What are you doing? It's okay. Lindsay just came home. It's okay. You can walk by. There you go. Very nice. All right. So it's always good to have a little break. Yeah, so that's what we do. And then with the COVID vaccine, it's an mRNA vaccine. But I've already talked about that, and that's really cool new technology. But in general, vaccines are when you give pieces of dead or just like pieces of the pathogen, and your body has the same response as if it was the real pathogen, but you're not going to get sick because it's not. All right, so where I'm wrapping up here. So a couple terms you need to know. Active immunity means that you've actually been exposed to this pathogen or a dead one or something like that, and your body has a response. And you make the B cells and the T cells, you make the antibodies, you make memory cells. Next time you're good to go. So active immunity means you got sick or you got a vaccine immunization. But passive immunity, as I talked about with MABs now, the monoclonal antibodies, is that you can take the antibodies that someone else developed and then purify them and then give them to somebody. And we do this, typhoid, you see tetanus here, rattlesnake venom, things like that. What we can do is we can take antibodies against that antigen or disease and give it to someone, and it doesn't last long, it lasts days or weeks, but it will attach itself to the antigen and it will neutralize it. But it's not long lasting, because then they're just cleared by the body. Like, you haven't activated your own immune system. Yeah. And then secondly, real importantly, early in life, more natural passive immunity is that early in life you're given antibodies from mom in the breast milk, and a few go through the placenta. But so you're breastfeeding. It's kind of the benefit of breastfeeding is that it's going to not only give the nutrition, but it gives antibodies from mom. And so the kid early in life gets a head start by having antibodies from mom, and those will wear off, and then they've got to develop their own, and kind of a critical period later on. All right. So this passive immunity, either giving antibodies on purpose or through breast milk, is useful, but not long-lived, like if it's active immunity. Yeah. So again, naturally acquired active immunity means you got exposed to that antigen and you dealt with it. And so now you have a memory. Artificially acquired active immunity is someone gave you a vaccine and you were exposed to it, but you didn't get sick because we gave you just a piece of it, not the whole thing. That artificially acquired passive immunity is when we give you antibodies. In the case of rattlesnake toxin, we shoot up a horse with venom from a rattlesnake, and the horse gets sick, it doesn't die. And then we take the horse blood, and then we take those antibodies and we dehydrate them in a little vial. We put it in a hospital. And if you get bit by a rattlesnake, we hydrate that, put it in your bloodstream, and those antibodies will stick onto any remaining venom and hopefully neutralize it. And then naturally acquired passive immunity is antibodies passed from mom to the baby. All right, your book talks a little bit about this. What about allergies, and what about anaphylactic shock? And so sometimes your response can be inappropriate. Your immune response is going to be out of proportion and inappropriate, right? A little ragweed, oh, I gotta have a stuffy head and runny nose for a month, right? I mean, that's not necessary, right? Or you eat a peanut and your body just closes up, you get hives, you're scratching. And so these are, inappropriate overreactions, you would call them. And they are. And what it is that you're kind of primed. Your body is primed so that these mast cells, that kind of these basophils that are in your tissues, they when exposed to something, an antigen like pollen or something, they will prime themselves, having all kinds of receptors. So the next time it comes, boy, it is just like on a hair trigger, and it will just release this histamine and heparin, and you'll have this big reaction taking place. Yeah, and so yeah, indeed, and so yeah, I think there's all kinds of directions I could go with this, but I think I'll leave it at that. Hypersensitivity reactions are called. Yeah, it's where your body is primed and then it way overreacts to a situation. All right, so we'll talk about, obviously transfusions with blood. We talked about the danger of getting the wrong type of blood. That has it. If I'm type B, if I get type A antigen blood, I'm going to antibodies will attack it, right? Because I have those antibodies, those anti-A antibodies I have because I'm type B, definitely. Transplanting organs, I get your kidneys, every cell in that kidney has got MHC1 showing a different type of signature than me, and my body's going to recognize that transplant as foreign and attack it. Yeah. Allergies, of course. And then I'm sure all of you are familiar with HIV/AIDS, but it kills helper T cells. Damn it, that's right in the middle. You need those to have an effective response. So what people die of is going to be infections that normally wouldn't kill you. You'll see people with HIV having fungus growing in their lungs or skin conditions that simple things that, it should not affect people with a nice immune system, but this virus attacks those helper T cells. Yeah, and again, if you're giving a transplant, the closer relative it is, identical twin would be great, but the further away it is, the more distant the chemicals are on the surface, and usually you're given a whole bunch of immunosuppressive drugs, to try to keep you from rejecting that transplant. And so some terms, so we talk about, this could be skin, like if you lose a piece of skin, you can take skin from elsewhere and put it there, or even someone else's skin, but you're going to reject other people's skin. But an autograft is your own from yourself. You take it from somewhere else, you put it there, you're not going to reject it, right? It just came from your butt to your arm, right? Like your body's like, that's cool, right? An isograft is nice. It's from a twin, identical twin. So yeah, so this could be skin. If you talk about like a leukemia, bone marrow, things like that. So that's your next best. Autograft is the best. Autograft, auto self, right? Isograft, good. Allograft comes from someone else. Yeah. So a kidney you're getting from a neighbor or something, or a friend or something like that is not related to you. And then a xenograft, xeno means foreigner, like xenophobic, because you don't like afraid of foreigners. This means from a different species. And we talked about heart valves from a pig. And so it's going to work, but your body is going to notice that it's different. All right, finally, big lecture here. But what happens with time? Well, your immune system, your immune response gets worse as you get older. Early on, I mean, it's all told by your thymus gland. Your thymus gland is big as a fetus, and it's big until adolescence, and then it shrinks and gets really small. And that's where you're going to mature these T cells. And so as you get older, you have less and less ability to fight infection. Technically, like the number of B and T cells doesn't really go down as you're older, but we know as you get older, you're less able to fight infections. And so we need to be particularly careful about that. Exactly. So when you look at not only pharmacologically, but surgically, candidates as they get older, that we know that they have less ability as young people to fight infection. All right, defense indeed. All right, class. Gave you a lot. So, from the lecture, you're going to study this chapter. You're going to hopefully take the terms that I've given you and understand the basics of T cells and B cells, humoral and cellular response, know the types of grafts, right? Just covered that. And know antibody structure, know what antibodies can do, know the difference between active and passive immunity. So, if I was studying, I would, make a study outline, make sure I know these terms, because I would predict they're going to be on multiple choice question next week. All right, good stuff. Again, the immune system, immunology. I admire the people that are immunologists. It's complex, but fascinating. And think of us humans having, mammals, having this ability, how infection doesn't, kill us every day, and how we heal, and how we have this memory of past diseases and be able to respond so quickly is just, really, really cool. All right, gonna sign out now.